Our presentation today is on bacteria and how they're named, where they live, what they do, how they reproduce, and anything you might need to know about the basics of bacteria. Bacteria are found in 2 of our 6 kingdoms. Either the eubacteria, these are the true bacteria, the things that you think of when you think of E. coli or salmonella, or the things that make you sick, and the archaeobacteria. These are ancient bacteria that live in extreme environments. Bacteria are microscopic prokaryotes, which means they do not have a nucleus. They lack a nucleus. And they are adapted to living in all environments, even some of those environments being very extreme. They basically exist everywhere. The structure of bacteria is fairly simple and pretty much the same for most bacteria. We'll focus first on the cell wall. The cell wall, which you can see is located underneath something else, right here, the dark black, that keeps the cell from bursting or collapsing due to changes in water. So if the bacteria were to be in an environment where there's suddenly less water or more water, the cell wall is what keeps it from bursting or shrinking. The cell wall might also be surrounded by an organized capsule called the glycocalyx, or by a loose jelly-like sheath called a slime layer. So that's what you see beyond the cell wall right here. This would be either the slime layer or the glycocalyx that in addition surrounds the cell wall. In parasitic forms of bacteria, this outer covering protects the cell from the host's defenses. So if you were to get bacteria into your body, of course your body is going to try to fight that as an infection. And the extra capsule around the outside of the bacteria helps fight against your defenses. Continuing on with bacterial structure. Bacteria have a flagella, which is used for movement. It's this tail that's right here. Allows the bacteria to move like you saw in the animation in the first slide. They have pili, or also called fimbriae. These help the bacteria cling to surfaces. These are found around the outside edge. Prokaryotes do not have organelles inside them or a membrane-bound nucleus. So you can see all of the internal structures are just kind of in one big mass. What they have instead is called a nucleoid region, which contains a circular loop of DNA. Even though in this photo or in this picture, it looks like the DNA is all jumbled up together, which would be what I'm pointing out there, it's actually one big loop. This ring of DNA, or an additional ring of DNA, is called a plasmid, and that can be used in reproduction. And bacteria have ribosomes in the cytoplasm, just like other cells, which are responsible for protein production. Just to review, the cell membrane, like in any cell, regulates what comes in and goes out of the cell, and bacteria are cells. So the cell membrane would be the innermost layer of membrane. Here it's called the plasma membrane. The cell wall is responsible for maintaining the shape or the form of the bacteria and keeping it from being destroyed in sudden changes of environment. And the capsule would be found outside of that, which is found in virulent bacteria and helps invade the immune system. On your assignment, question number 8, what are these 2 blank areas called? What is this pointing to? And what is this structure here? If you need to, go back in the video and listen again. Let's move on to how prokaryotes reproduce. The main form of reproduction is called binary fission. If you look in the photo here, you can see that the bacteria is making a copy of its DNA. So we start with one green bacterial DNA loop, and then it turns into the pink and the purple, which happens the same way as it does in regular cell reproduction, cell division. It goes through a process of mitosis, it goes through a process of DNA replication. So you have 2 copies of the same DNA, and then the bacteria simply splits. Binary fission is known as the splitting of a parent cell into two daughter cells. This would be asexual reproduction, meaning there's no crossing over of genes, there's no sharing of genetic information, and it happens in prokaryotes, which are things without a nucleus. Like I said, basically DNA makes a copy of itself, which is called DNA replication, and then the cell splits apart. There are, however, ways for bacteria to share genes among other bacteria. There are three ways, notice in this picture here. Transformation. The important part here is this little purple gene. Conjugation. And transduction. We'll learn about each of these three, starting with conjugation. Here you see a photo of conjugation. This occurs when a bacterium passes DNA to a second bacterium through a tube called a sexpillis. You can see right here, there would be a tube connecting them, or you can see in the actual photo, the tube connecting the two. The DNA gets passed between one bacterium to another. This temporarily joins the two cells, and it only occurs between bacteria in the same or closely related species. So two unrelated bacteria could not have conjugation. Another way for bacteria to share genes is through transformation. This involves bacteria taking up free pieces of DNA secreted by live bacteria or released by dead bacteria. So if bacteria suddenly die, for whatever reason, their DNA becomes floating in the environment of other bacteria, and in transformation, the living bacteria can simply uptake, probably through phagocytosis, like we learned about earlier. And that bacteria just becomes part of the living bacteria's, that DNA becomes part of the living bacteria's DNA. This is how transformation works. The donor DNA fragments bind to proteins on the cell surface. Here's the bacterial cell that's going to uptake this DNA. Then the donor DNA fragment is transported into its host and becomes single-stranded. In successful transformation, the donor DNA is incorporated into the host's chromosome. Remember, in bacteria, that's one loop of DNA. If it is not successful, then the donor DNA just simply degrades and gets absorbed into the rest of the cell. One final way that bacteria can actually share genetic information is through transduction. This requires the use of a bacteriophage, which is a type of virus. Virus is a non-living thing, to transfer portions of the bacterial DNA from one cell to another. So here we have the bacteriophage. It attacks this bacterial cell. You can see that if you look closely at the DNA in the virus, it takes up part of the DNA from the bacteria. The bacteria dies and releases all of the viruses that go to another DNA host or another bacterial host and inject the DNA from the first bacteria into a new one. Plasmids are used in transduction. These are separate pieces of DNA that can replicate on their own, and they can carry genes for resistance to antibiotics and transfer them between bacteria by any of these processes. So transduction is, in the medical world, not a good thing because this allows bacteria to become resistant to the antibiotics that we try to use to fight them. Again, a plasmid is an extra little bit of DNA, like you see here, and it's used in sexual reproduction of bacteria. They can also be used in genetic engineering. Some bacteria form resistant endospores around the outside. It's a hard covering that keeps the cell in hibernation until a later time in response to unfavorable environmental conditions. Moving on to prokaryotic nutrition. So how do bacteria get the nutrients that they need to survive? You can see here that this is a photo of Staphylococcus, which is a gram-positive facultative anaerobe, meaning it does not need oxygen for its survival. Bacteria are very different in their need and tolerance of oxygen. Some of them need oxygen, some of them cannot tolerate the presence of oxygen. The first type of category is the obligate anaerobe. These are unable to grow in the presence of oxygen. This includes anaerobic bacteria that cause botulism, gangrene, and tetanus. And as you can see down here. And then facultative anaerobes are able to grow in either the presence or absence of gaseous oxygen, like you see in this photo. So obligate anaerobes, they are obligated to not have oxygen. Facultative anaerobes can use it, but they don't have to. And then aerobic organisms. These include animals and most prokaryotes. These require a constant supply of oxygen to carry out their cellular respiration. When we talked before about cellular respiration, we also talked about fermentation. These obligate anaerobes, and probably some of the facultative anaerobes, would be going through fermentation instead of cellular respiration as their main source of energy producing chemical process. Continuing with prokaryotic nutrition, we have autotrophic prokaryotes. These mean they feed themselves. They can either be photoautotrophs. These are photosynthetic, like plants. They use light energy to assemble the organic molecules that they require. Or you can have chemoautotrophs. These make organic molecules that, by using energy derived from either the oxidation of inorganic compounds in the environment, like methanogens, and you have thermoacidophiles. These would be archaeobacteria. They thrive in super hot, super acidic condition, and in this photo here it would be the red stuff. You can see that's a massive amount of bacteria. And these are more recently classified in our Six Kingdom system. You also have heterotrophic prokaryotes. We just learned about autotrophic. They can feed on, they make their own nutritional molecules. But there are prokaryotes, there are bacteria that require other things to feed on. Most free-living bacteria are chemoheterotrophs that take in preformed organic nutrients, meaning they feed on other living or decaying things. There are probably no natural organic molecule that cannot be broken down by some prokaryotic species. These are the things responsible for breaking down everything that dies. The name for this bacteria is detritivore, or saprophytic. These are critical in recycling materials in the ecosystem because they decompose dead organic matter and make it available to photosynthesizers. So basically, something out in the forest dies. It's the detritivores, which would be saprophytic bacteria, that break it down, using that dead organic matter as their own source of nutrition, turning that, plant or animal into something that the plants are going to use or going to get their nutrition from. Plants, of course, make their own sugar molecules through photosynthesis, but they need other things from the soil in order to continue that chemical process. So bacteria have a very important role to play in breaking down materials in the environment. We could not live in our current ecosystem without the role that bacteria play. However, some are harmful and break down material that we'd rather keep, like this image of an infection of necrotizing fasciitis or flesh-eating bacteria. So there's pros and cons to heterotrophic prokaryotes. Now let's talk about bacterial shape and naming. The Gram-stain procedure, which was developed in the late 1880s by Hans Christian Gram, differentiates bacteria. You can see here there's two possible outcomes through using the Gram-stain procedure. This would be done in a lab or in a science classroom to differentiate one bacteria from another. Gram-positive bacteria stains purple, whereas Gram-negative bacteria stains pink. So in the top photo, these would be Gram-positive and these would be Gram-negative. The difference is dependent on the cell wall of the bacteria itself. If the cell wall is thick, it becomes gram-positive. It soaks in all of the purple stain that they would use in this little procedure and shows up purple. If the cell wall of the bacteria is thin, it doesn't soak in as much of the stain and it appears pink when you look under a microscope. So here, gram-positive turns purple. This is the big thick cell wall, and gram-negative appears pink. This is the thinner cell wall. So these photos of bacteria are gram-negative, they're pink. And these would be gram-positive. In just a moment, you're going to be asked to identify whether the bacteria in the photos are gram-positive, gram-negative. This, gram-positive or gram-negative? That would be gram-negative, as it appears pink. Although this is a SEM photograph, so they could have used other stains too. The gram staining procedure is used under a light microscope. Now let's move on to bacterial shapes. There are 3 basic shapes. The cocci, these are the spheres that you see in this photo. The bacilli, these are the rods. And the spirilla, which would be the spiral shaped. There's an additional naming system. You either have staph, which means it's found in clusters, or strep, which means it's found in chains. Remember those 5 things. You're going to be asked to name some things as we move along. So staph is found in clusters, strep is found in chains. Streptococcus is in the image here. This is the bacteria that causes strep throat. Coccus means spheres. Strepto means in chains. So you can see here you've got spheres that are linked together in a chain-like form. That would be Streptococcus. That's how you name that. Okay, now for some identification. Identify the photo as gram-positive or gram-negative, and then identify the shape of the bacteria as Bacillis, cocci, or spirilla. This appears to be gram-negative because it's pink. And the shape of the bacteria is in a rod, which would make it a bacillus. Now identify this as gram-positive or gram-negative. And the shape, is it Bacillus, cocci, or spirilla? And the photo you're seeing here is Anthrax. Is this gram-positive or gram-negative? Now, identify this as Streptococcus. We know it's coccus because it's in spheres. Or is it Staphylococcus? Streptococcus would be found in chains. Staphylococcus would be found in clusters. Strains of this particular bacteria are responsible for strep throat and flesh-eating bacteria. Identify this as Streptococcus or Staphylococcus. Incidentally, in Greek, the term staph means bunch of grapes. So that hopefully can help you remember whether it's found in chains or clusters. Identify this as either cocci, bacillus, or spirilla. This particular bacteria is easy to grow in the lab and not harmful. It would be gram-positive, so it appears purple. However, this is an example of a poor stain because in the stain you can see it looks both purple and pink. Gram staining takes practice. Let's talk about some specific types of bacteria. First is tetanus. Tetanus, if you were to be infected with tetanus, also known as lockjaw, causes rigid muscles. You can't move your muscles. They clench up, harden up, and makes you unable to move them through the action of this bacteria. Clostridium botulism is a gram-positive bacteria that causes food poisoning that can sometimes be fatal. All forms of botulism lead to paralysis that typically starts with the muscles of the face and then spreads toward the limbs. In severe forms, it leads to paralysis of the breathing muscles and causes respiratory failure. In view of this life-threatening complication, all suspected cases of botulism are treated as medical emergencies, and public health officials are usually involved to prevent further cases from the same source. However, we also know that botulism, or a strain or form of botulism, is used in Botox treatments as it paralyzes the face muscles and effectively smooths out wrinkles, but as we know, results may vary. Another nasty bacteria is Yersinia pestis, or Y. pestis. This is also known as the Black Plague, and this bacteria was carried on the fleas found on rats. It was responsible for the death of thousands of Europeans from the 8th to the 14th century. And Bacillis anthracis, or anthrax. This infects mostly farm animals and is usually spread to humans through some sort of break in the skin. E. coli. Before we move on, what is the long thing at the end of each bacterium called? One of these, or these, or these. Is this bacteria cocci, Bacillis, or Spirilla? And Salmonella. Here's an animation of Salmonella swimming. Name this bacteria. Is this Staphyl or Strepto? And then add to it the second part. Would it be Staphylococcus or Streptococcus, Staphylobacillus or Streptobacillus, Staphylospirilla or Streptospirilla? Culturing bacteria requires some nutrition in the form of usually a plate, but sometimes in a liquid. And once you cultured the bacteria from a particular source, like you see here, then you could take it, put it on a slide, gram stain it, put it under the microscope and identify it. And just a little humor. If you have any questions about this presentation or the assignment that you are to do with it, please don't hesitate to message me.